The overall goal of this stereological study using the optical fractionator is to obtain quantitative information, such as the total number of cells in a specific brain area, using methods based on unbiased principles.
This method can help answer key questions in the field of quantitative analysis, such as estimation of the total number of cells in a complete structure of interest.
One main benefit of this technique is that it provides accurate estimates with a fixed and predetermined precision.
Demonstrating this procedure is Esther Kjær Needham, who is a co-author of the article.
Begin with a fixed and frozen rat brain. Using a scalpel, separate the right and left hemispheres along the midline of the brain, and then choose one or the other hemisphere randomly in the first brain. Thereafter, shift systematically between right and left hemisphere. Then use mounting medium to mount the chosen hemisphere onto a specimen disc with the long axis perpendicular to the disc. When the hemisphere is mounted on the disc, place a tube around the hemisphere and fill it with tissue tech to completely cover the hemisphere. Next, place numbered multi-dish containers into the cryostat for pre-cooling. Then mount the specimen disc into the cryostat and cut the entirety of the hippocampus, starting randomly, into 80-micron thick sections in the coronal plane. While cutting, place all sampled sections consecutively in the cooled multi-dish containers to ensure that the sections are kept in cutting order. When the whole hippocampus has been sectioned,